Hey, we're live. We're group 4. We're going to do lesson 13 problems. I'm Keegan.
Yana.
Penny.
And I'm going to do question 1. So I'm going to pull up my screen and then we can start sharing. Okay, so yeah, this is the lesson 13 problems. So this one it says you wish to use the DNA sequencing Sanger method to determine nucleotide sequence. Has a lot, it gives you a sequence. So I copied that sequence down here and then I made a complementary strand below it and then used that strand to make my gel. So you basically just go in order. So it's A, T, you can't see under there, but it's A, T, C, A, G, G, T, T, A, C, G, T, C, and that just follows this order. And the X's, it just represents like a strand on the gel. So that's what your gel would look like if you did it in the positive electrodes down here at the bottom. For B, it just gives different scenarios and asks what would happen if this was added. So it says, suppose I use E. coli DNA polymerase during sequencing reaction instead of tag polymerase. And this wouldn't work because DNA polymerase would denature. Tag polymerase is heat resistant, so it works with PCR reactions, but any other polymerase would just denature and not work. For C, it says, suppose I accidentally add E. coli DNA helicase to the reaction in addition to the normal DNA sequencing reaction components. And this reaction should just happen like it normally happens. Helicase usually separates strands, but heat does this in PCR. So the helicase would just be worthless and would just be there. And for D, it says, suppose I add DDATP to one of the 4 tubes. However, I forgot to add DDGTP, DDTTP, and DDCTP to the other tubes. Assume that the other components used for the sequencing are normal. So what would happen is the termination would just occur when the T base is read by the DNA polymerase. And yep, that's question 1.
Okay, and so for A in question 2, it's just the vocab that we got from the reading. And so during a typical PCR cycle, the synthesizing or the DNA synthesizing step is done at 72 degrees Celsius, while the annealing step is done at 55 degrees Celsius. For B, AMP-R is a common selectable marker that is found in many plasmid vectors, while restriction endonucleases recognize and cleave DNA molecules at palindromic sites. And then C, during agarose gel electrophoresis, DNA is placed in an electrical field. DNA migrates towards the positive electrode during electrophoresis.
So for D, we have DDATP lacks the 2' hydroxide group and the 3' hydroxide group. For E, transformation is a term used to describe the process where bacteria take up DNA molecules from the environment. And F, during the blue-white screening, IPTG is commonly used to induce the expression of the lac operon, while X-gal is cleaved by the beta-galactosidase to turn E. coli cells blue.
Okay, and then for question 3, A asks, write the single-stranded DNA sequences that you would use for the 2 primers used in the PCR experiment. Note that each primer should anneal to one of the sequences in bold. And so for the first primer, also known as the forward primer, you're going to start at the top, in the top left corner, the 5'. And so we found GAG, CC, TT, CAC, T, CAG, TC, and it goes 3 to 5. Or sorry, 5 to 3'. And then the reverse primer, it's going to go 3' to 5'. And so on the right-hand corner, the 5' ends. But reading from the 3', we have TC, GGA, GTT, AAC, GTG, ACT. Right?
Yes.
Yes. Okay. And so those are our 2 primers that we found for A. And then B, it asks for the primer temperatures, correct? The annealing temperatures?
Yeah.
And so we use the equation above. And so you take the 81.5 and you add 0.41. and you're gonna multiply it by the amount of bases, the GC by the percent of GC content. And so for the first primer, it's 56%. And then you're going to subtract by 675. I believe that is the total number of bases. And then you're going to divide it by N, which is the nucleotides, which is 16. And then we get 62.4 degrees Celsius. And for the second primer, you do the exact same thing, except for the percent GC content, we have 50 instead. And so we'd find 59.8 degrees Celsius. And then for, I think our lettering is a little off.
Yeah, this is C, sorry.
Yeah, no problem.
This is C.
So C, as for each PCR cycle contains a DNA synthesis step. What are the sequences of the two newly synthesized strands if DDCTP is accidentally included in the PCR experiment? And assume that DATP, DTTP, DGTP, and all other appropriate PCR components are found in the reaction mixture. So if you'll go back up, Keegan. So for the first one, we start at where we found our first primer, but we start at the end of it. And so we find GGG, and then ATT, AGC. We're going to stop right at that C because that's where the DDCTP stops it. So we included the C because it's accidentally included, so I'm not sure if it's left out, but we included the C. And then it's the same for the other, but it's on the right hand where we had our reverse primer. And so we found CT, oh, sorry. So we're going to go 5 to 3 prime. So we start TTT, TTT, oh no, sorry, but it's written 3 to 5 prime still, but you read it 5 to 3, so it's TTT, AATC, and then we stop at that C as well. Sorry, that was a little confusing.
No.
And then for D, so we found that DAM is not included in a PCR because it needs continuous replication, and it would stop that because it stops methylation. And then DNA A, it catalyzes the separation of the two template strands, and so it isn't needed in a PCR because the heat performs the separation during the denaturing step.
Awesome. All right, for number 4, so this question concerns the agarose gel electrophoresis and restriction mapping. Suppose you set up 6 restriction enzyme digestion reactions. Each reaction contains the same linear DNA molecule, but in each case, the DNA molecule is digested with different combinations of the restriction enzymes. BAM-HI, is that it? Yeah. EcoRI and HIND-3. These reactions produce the following results. So the first question, after restriction enzyme digestions, the contents of the 6 reactions are loaded into separate wells of an agarose gel and are subjected to the agarose gel electrophoresis. Draw an image that represents the result of agarose gel electrophoresis. For this problem, assume you receive 1 lane of the agarose gel from a marker sample that contains 1,000 base pairs, 100, 200, 300, 400, 500, and 1,000 base pair fragments. For the drawing, assume that the positive electrode is at the bottom of your drawing while the negative electrode is at the top. So in the far left column, we have our base pairs. And the next column is our sample with the markers. And everyone gets one because that's what the markers are. And so then if you look at the results in the table above, the corresponding X's correspond with the number of base pairs for that enzyme. So for BAM-HI, we get 500. 500, I can't see it, I gotta look, but it's like I think 350 and 500, and that's where those are marked on this table. Same for EcoRI and HIND-3, and same for the combined ones as well to generate this banding map. All right. So draw a linear restriction map that indicates the location of the restriction enzyme sites along this hypothetical DNA molecule. So we have it at 50, 150, 100, 100, and 500. So you have the BAM-HI, EcoRI, HIND-3, and BAM-HI respectively. what I guess I'm like, shoot, because she likes us to really demonstrate how we did this, and we watched her video how to do this. It was really complicated.
She explains it better.
And it took 8 minutes. So, but yeah, that video was really helpful for the mapping, because I think the group problem question is a lot harder than the practice problem. So what are the predicted fragment lengths for this linear DNA molecule? It's cleaved with all 3 molecules simultaneous. So we get 50, 150, 100, and 500, and the 100's bolded here because it would be a thicker strand because there's 2 of them, and that is why we bolded that there. Tanya and Keegan, do you have anything to add for that one?
No, that sounds good.
No, that's good.
Tatiana.
Okay, and so I have 5. So it says, you're interested in studying a gene called your favorite gene, YFG, that you have identified in the yeast Saccharomyces cerevisiae. Detail the steps that you would use to generate cDNA from Saccharomyces mature mRNA, clone a cDNA version of YFG into a plasmid vector, and using blue-white screening to identify plasmids that contain the YFG cDNA. So to generate the cDNA, kind of just a quick explanation. So we would purify the mRNA sample, then we would mix the mRNA with the primers. We would add reverse transcriptase and the dNTPs. We would add RNase H to digest the mRNA, add DNA polymerase from E. coli and DNA ligase, and that synthesizes a new strand, and then they're cut by the restriction endonucleases. To clone, you'd take that cloning plasmid, the sticky ends of the plasmid would line up with the sticky ends of the DNA. They would come together and phosphodiester bonds would be formed by the DNA ligase. Transformation incorporates into the host cells, the E. coli cells, and this would form new YFG cells. And to do blue-white screening, you add IPTG and XGAL. If they turn out blue, then lag-Z happened, and that's done with the beta-galactosidase. And if they turn out white, then transformation was a success. And that's what you know happened if they turn out white. So yeah, that is our video. If you have any questions, or do you guys have anything to add?
No, it's good.
Okay. Well, sounds good. I will stop it then.